Hello. Previously, I presented to you all how to write scientific writing and it was giving you the tool inside how exactly a research article is all about and how to write it down. So on this part, I'm going to explain it to you on how to write materials and methods section. As I have explained to you before, that this material and methods section is the most easiest part, and you can start from this sections if you find it very difficult to start writing your publication. So as materials and methods, it seems that's the most easiest reading section, but it's most important section, or the crucial section. Why I say it is very crucial is that it is, as many of the scientists, what is important in science is that it's reproducible in results. So every time you perform the particular experiment in the same way how you described, it should be performed by the other scientists in the same way as you described, and see whether that protocol works or not. So that's very important. Second thing, I say that one should start writing with this section first. This is basically, as I told you, it's the most easiest section, so you think you'll finish something faster read and you'll say like you accomplished, oh my god, I've written manuscript, materials and methods, and I'm feeling better right now, so I can move on to the next section tomorrow. So that gives you a boost so that you can continue doing your writing work. Next thing is that in spite of being such an easier section, the most of the scientists reject your manuscript because of the materials and methods. There are around 30% of rejection of the manuscript due to this section. Why do you think so? Because this is the core part of your entire research article and will tell you whether your manuscript is reproducible, whether it's important to the science, whether something new has come up to this, or is it the same thing which is going to be described in this particular manuscript, like how other people have described. So it's nothing new to the science. So if they think all these particular questions in their mind, the reviewers will keep thinking, or the editor will think, and they'll reject it. So I need to write correct materials and method. First of all, I said that it should be reproducible, so that it should show the transparency to your research, what you're performing. It should validate your designs, whether the design which you used for that particular experiment was good or bad, or to understand if the study can be repeated or not by the various scientists across the globe. So what you have to include in this particular section? You have to explain the different methodologies you have conducted, what you did and how you did it. That's what you have to write step by step without missing anything. The most obvious thing that you used is not to be written. It's like some things are common sense which can be avoided, but some things you have to mention. Like you have to mention the standard methods which you have applied, whether you have quoted particular method which you have used from the other researchers, or you have designed your own protocol or method to conduct a particular experiment. You have to write it down on what basis you have chosen a particular step in that particular method. Like maybe, for example, you've seen that your product has protein bounds to it and you want to remove the proteins from the product, so you might use the protein denaturation temperature, for example. So that step was missing with the previous researcher maybe, so you've incorporated it by doing some of the observations. So certain things you have to mention and why you did it and how you did it, and write it down whether you have done it or previously someone else had done it. So that will give you a new position for your manuscript. Then you have to mention your manufacturing locations of mostly what product you have used, like such as reagents or chemicals you have used, or any standards you have used that you have to write, or from where you have purchased it, which company, location, because this will help for the other researchers to exactly to go and pinpoint what chemicals they have used and what results they have obtained, whether they clash or whether the same results, whether using the different industrial products, why there are some changes. So that also can be understood, because there is a lot of difference the type of reagents or chemicals we use also, because certain companies, when they design the particular product, it is said that it has universally the same thing, but there is a change from batch to batch, as every pharma company knows. Like what happens is that, for example, I'm growing a cultures on malt extract agar, and I use a malt extract powder which I'm obtained from, for example, maybe High Meteor or any other company. What will happen, there will be malt which was used in particular, making that particular batch might not be the same quality or product you obtain in the next batch. So that possibly there will be changes in the nutrition quantity. They do say that particular things are calibrated, but there can be possibility of 0.01 gram also. So that difference can be observed by various bacteria or fungal growth, or any yeast which will be growing on that particular medium, because they need nutrients in 0.01 gram or milligrams of changes can also make a lot of difference in the particular results. So what will happen is that there might be some changes, difference, like such as colony growth diameter will be different, maybe the aerial growth might be different, or the colony textures, the growth rate will obviously differ when there is a change in the product by 0.1 centimeter or millimeter difference. So that's important to note down what reagent you had used, when it was done, that all counts. How data analyzed is the next thing. The data can be analyzed by using various statistical test methods. So you have to write it down which particular test you had used, on what parameters you had applied for that particular analysis to be performed. Now how to write? You have to write first of all in details as by whatever points I mentioned to you. But the thing is, you have to write it in the past tense or in passive voice. This is the most important thing. You have to use verbs like investigated, evaluated, performed, and you should avoid inactive voice. You have to present your data very logically by mentioning which date it was collected, your survey which was performed, or sampling size or method which was used. You have to also mention the time and year which the study was carried out. Or maybe it was seasonal, or it is throughout the season. So you have to understand its phenology also. So data collection time and survey stats are very important. You have to also mention whether the evaluation or exclusion criteria that you have been used in this particular study. For example, exclusion criteria like a particular data set you have not included in this study. Why you have not included, you have to also mention. Now, the next thing is you have performed your experiment as per your time, and like simultaneously you carried out 2-3 experiments on the same day, but you're not required to mention it in chronological the way you performed it, but you should be more sensible how the paper needed. Like you have done all the experiments, but then you're not necessary to write what you have done on the same day, but it should be like how exactly that full protocol completes. That's important. Tables or figures to summarize. You can use certain tables or figures in your materials and methods is to give it much concise understanding to your materials and methods, or you can avoid it also. And do not fail to give captions to your tables and figures, which is very important. Always use a standard units of measurements or SI units, and international abbreviation should be used across wherever possible, and these abbreviations should be explained within parentheses at the first mentioned in the manuscript, and later you can just use the abbreviation and do not need to write it your full form of it again and again. So now we'll take some examples for understanding. I'll give some examples which I have in my own published papers, wherein you can understand how the material method section is written and what it should include, like whatever points I mentioned, and based on that you'll get an idea. So if we go to the first example, here is this paper, you can see that we'll go directly to the materials and methods section as we are dealing with only materials and methods. In this particular section, when there are subsections which I've created, so the subsections, like first of all was the source and growth condition of the melanic cultures, like what was my source which was used for that particular study. Secondly was how it was used, like, and to do what was the next question, and what are the different methods which I have applied to obtain that particular melanin from that culture, and the different instruments are used to study that particular melanin from that mushroom or any organism. Okay, so this is how the materials and methods were written. Now we'll go to this source, where you have to mention what type of cultures, from where you have obtained it. The location is very important. Collected is all in past tense, you can see. So, and this is the location you have to maintain. How you had used it, so you have to give the, like you have compared this with some standards, and then you came to know that for a particular species you matched with that. The medium which was used, you can see here the full form, and then the universal abbreviation is written to the parentheses, and then whenever you require it, we use the same abbreviation in the further part of the manuscript. You have to also mention the type of grade you use, or the amount of substance which you have taken. The unit should be proper, and the chemicals from where you have obtained and which company you have to also write it down. So that's what I was mentioning previously. So whatever media you have used, you have to give its detail, the incubation and all that comes from your source. And then how the method, how to grow this particular culture, like I said, the instruments details you have to give. So like I use a rotary shaker of what make it was used. I have already mentioned in the bracket, Architect model, which one, from where it was purchased, etc. Right? Like if you use a standard methods, now for example, the mycelial suspension was made by using a standard published method, where so Khalis et al. In 1986, that method was not necessary that you need to describe it all over again. You can just mention it in the bracket that this method was applied. Then you can, the next thing what you can see is like this is a detail protocol I have given, because I've said that this Sun et al. Method which was used for extraction, I have been modified it, so I am using modification in the previously described method. So then I have also mentioned modification in this particular, which is the temperature and the concentration of the NaO which was changed. So that was what is mentioned. And the various instruments which were used, the studies, whether it was done in your own laboratory or outside, outsource the product, you have to also mention it and make it clear in the particular publications. So for example, now we can see in this bright field microscopy, we have to use particular microscope, that is Nikon, and then what camera it had used, that also, and what software used for image analysis have been also mentioned. The second thing is that the images, when we taken, how they are processed, it has to be also be mentioned, because certain magnification will also interfere into the study outputs results. So like if it was used 10,000x, then you have to mention 10,000x magnification, same images were used, and what software you had used, which version you had used it, and when it was available, and from where you got it can be also be mentioned to give a more detail, so that the people can use the same software and get your results. Okay, so if that this work was outsourced, then you have to also mention that this was not done, it was done by someone else. So then it is here mentioned it was done at SAFE IIT Bombay Analytical Laboratory in India. So certain things were outsourced, so that has been also be mentioned. So here we finish the materials and methods in example 1. We'll go to the example 2 for y'all. Now in this you can see material method section. Okay, here it has again collection of fluid bodies, the details you have to mention, what sources you had used, the whatever names were given. I have given a short form for this particular thing. Culture, these are not universe, but these are assigned to the particular culture. So this throughout I do not need to use the microcarpus or albuminous names, so I can use this as a short form abbreviation. Okay. And next thing you have to coming to this example to show a statistical analysis. Like when you perform a statistical analysis, that also we need to mention in detail how you had done it. Like 1B ANOVA analysis was carried out by using Turkey on a significant HST test, and that was applied in using the Excel statistical software with the package version of 2016-7. As software keep on updating, so I have to mention when I used and which version also is important. And when you perform particular graphs and all, like how you performed it was using my Systed software, and this details have to be mentioned so that the other person who is going to do that analysis can perform the same statistical analysis and produce the results if possible. Okay, so here I hope you got an insight to the materials and methods and understood the details, how to write it down, and key points. So thank you.